Animal models are used for addressing many scientific discoveries, for example, drug discovery, drug screening, but many times it is found that the result generated by animal model cannot be directly translated in human model system. So as a result, pharmaceutical companies or cosmetic companies use huge amount of time and money for it. Region Artists Private Limited is trying to develop artificial human skin by 3D bioprinting that can be used for addressing many scientific purposes. It can be used for drug screening applications, and it can reduce unnecessary use of animal models. So pharmaceutical companies and cosmetic companies are developing lot of drug molecules. For that purpose, they are either culturing in animal model or in monolayer culture. But monolayer culture and animal model do not represent the complexity of human skin system. So in 2015, European Union had complete ban on using animal model for cosmetic testing, and followed by European Union, many other companies have also implemented ban on animal model system, including India. We are trying to develop hypertrophic scar, psoriasis, and as well as keloid scar model, aging skin model to replicate anatomical feature of human skin. And then we are testing the drug molecules. Compound annual growth rate is 12.8% for cosmetic and pharmaceutical sector, and skincare segment will hold 26% share. So there is huge potential for this type of model for applying in cosmetic sector. We are trying to control orientation of cells and extracellular matrix by 3D bioprinting, and at the same time we are conjugating pro-inflammatory cytokines or anti-inflammatory cytokines, and then we are developing hydrogel, and we are mixing human cells to replicate the anatomical feature of human skin. And as a result of adding pro-inflammatory cytokine and anti-inflammatory cytokine, we are trying to replicate feature of specific disease condition. We are trying to replicate anatomical orientation of human cells as well as extracellular matrix, and at the same time we are chemically conjugating pro-inflammatory cytokines and anti-inflammatory cytokines to represent the tissue anatomy and pathophysiology. And we are trying to replicate the undulated morphology between dermis and epidermis of human skin, and as a result we are trying to simulate the anatomical feature as well as pathophysiology of human skin. We have got BIDEX support, and we are also conjugating with different pharmaceutical companies to lead this discovery.
I have loaded a CAD-based design in this computer. Now I am giving the command to the 3D printer to print that structure into our desired design. Now the instrument is printing, it moves in XYZ direction and build the structure into that predefined design.
To print skin tissue constructs, we are using our proprietary silk fibroin gelatin bioink mixed with 3 different types of skin cells, namely fibroblast, keratinocytes and melanocytes, along with different growth factors and differentiation factors. This combination is very much essential to recapitulate the undulated morphology of human skin.
After printing the 3-cell composition bioprinted skin construct, it is essential to check the immune compatibility and biocompatibility of the printed construct. For that purpose, we are using our live cell microscopy setup. In these systems, we are working with the primary monocytes, which we are isolating from the peripheral blood of human, and after culturing the monocytes, we are converting the monocytes in particular 3 setup. One is pro-inflammatory and one is anti-inflammatory setup, and co-culturing this particular pro-inflammatory and anti-inflammatory monocytes with this bioprinted construct will help us to get the knowledge of the immune compatibility and biocompatibility of this printed construct. So after implantation in human body, how this construct will behave and how this construct will respond, this knowledge we will able to provide before implantations. You will transfer me the construct, then I will check the biocompatibility by using this live cell.